Okay, welcome to Let's Get to the Marks. I'm going through the AS-Biology Paper 2 for 2021. This is for AQA. Make sure you pause the video at the start of each question, grab a pen, grab some paper, grab a calculator, have a go at the question, and then unpause and watch my explanation. All right, guys, let's get into it. Question 1, point 1. The general structure of a fatty acid is RCOOH. Name the group represented by COOH. Pause, have a go. All right, I'm going to go through the answer. This is known as a carboxylic acid group or a carboxyl group. And this is how you draw it if you wanted to display it like that. And here we go. Easy peasy, lemon squeezy, 1 mark. Let's look at question 1.2. Figure 1 shows the structure of a fatty acid R group. So you can see we've got carbon atoms, hydrogen atoms, and we've got a double bond hanging around there. Name the type of R group shown in figure 1. Explain your answer. Once again, why don't you pause the video, have a go, and then unpause and watch the explanation. Okay, what type of R group is this? Well, we've got to ask ourselves, what is a fatty acid? What's a fatty acid attached to? Well, fatty acids come attached to glycerol in triglycerides or in phospholipids. You get 2 fatty acids in phospholipids and 3 of them in a triglyceride attached to the glycerol. So this is the fatty acid part, which is this part I'm circling here in a triglyceride. Okay, so you've got these 3 fatty acid tails and you've got the glycerol group here. So how would we describe this type of fatty acid group? Well, let's have a look at the unique structure, and that is a carbon-carbon double bond. So we would say this is an unsaturated fatty acid. So the answer is unsaturated, and the explanation or justification for this is it contains a carbon-carbon double bond. You can just write that carbon-carbon double bond, or you could just put C with the equal sign and another C. Okay, and that will get you 2 marks. Right, we're going to move on to question 1.3. Describe how you would test for the presence of a lipid in a liquid sample of food. So what they're saying is the food has already been blended up, and how would you actually test that blended sample to see if there were any lipids in it? Well, you would shake with ethanol, and then you pour immediately into cold water, and what you see is a milky white emulsion. Basically, at the top of your test tube, you pour your sample into a test tube of cold water, you get like a milky white color towards the top of the tube, and that's called a milky emulsion. And this is known as the emulsion test. Okay, let's look at the next question. In 1935, scientists suggested a model for the chemical structure of a cell surface membrane. Figure 2 shows the membrane structure as the scientists suggested. So we've got this layer of proteins, and then we've got these. Hopefully you recognize these molecules here with the 2 tails and the round circle at the top. This is a phospholipid. Ah, you were going to say that. And so they've got 2 layers of that with the heads facing out, tails in, and then another layer of protein. Give 1 similarity and 2 differences between the membrane structure shown in this diagram and the fluid mosaic model of a membrane structure that we now know today. So what's the similarity? What do they both contain? Well, they both contain phospholipids. And in fact, if we go a little bit more detailed, they both have a phospholipid bilayer, 2 layers of phospholipids. And also what is the same is that the hydrophilic heads face outwards of the cell and to the inside of the cell, so towards the tissue fluid and then towards the cytoplasm because they're hydrophilic. And the hydrophobic tails face towards each other, away from the inside of the cell and away from the outside of the cell because they're hydrophobic. They hate water. So that's the similarity. Now let's look at some of the differences. Okay, so I would look at where the proteins are, have a look at where these the layer of proteins are. Do we normally see with a cell membrane with our current model, do we see a layer of proteins outside of the phospholipids? Where do you find the proteins in today's model of the cell surface membrane? That's what you've got to ask yourself. Where do you find those proteins? And hopefully you realize you find the proteins embedded within the phospholipids, either as glycoproteins, cell signaling proteins, or channel proteins, transporter proteins, carrier proteins for active transport, channel proteins for facilitated diffusion. So we find the proteins intrinsic and extrinsic there, but whether they're intrinsic or extrinsic, they are within the membrane. So you have intrinsic ones that run all the way through, like channel proteins. You have extrinsic ones like glycoproteins and sort of signaling proteins that are on the outside of the membrane. Okay, but they're normally embedded within the membrane. Now, there's another structure. What is it that regulates the rigidity or the flexibility of the membrane that we know in the current model that isn't in this model? And that's cholesterol. So if you notice here, they don't have any cholesterol between the the phospholipid tails, right? The hydrophobic tails. Normally, there's cholesterol floating about that regulates the rigidity or flexibility of the membrane. Okay, let's go on to 2.1. Describe and explain one feature of the alveolar epithelium that makes the epithelium well adapted as a surface for gas exchange. Do not refer to the surface area or the moisture. Moisture is good for gases to dissolve, increases their ability to dissolve, so increases the rate of diffusion. And surface area obviously makes a larger available space for diffusion. Well, we can talk about the fact. Do you remember what these cells are called? These are the squashed or flattened cells. We know them as squamous epithelial cells. And the epithelium made of these flattened or squashed cells is thin, right? How many cells thick? You all know how many cells thick the walls of the alveoli are, or the walls of the intestines. They're one cell thick. There's many exchange surfaces where the cells will be one cell thick. It's a great adaptation to make for rapid diffusion. So how does being one cell thick or having a thin layer or having flattened or squashed cells help? So it helps by creating a short diffusion distance, or you could say a short diffusion pathway. So it's not one common mistake is that people will say they have a thin cell wall. Of course, animal cells don't have cell walls, so that should immediately trigger alarm bells in your head that you're on the wrong path. We're talking about the tissue, the whole structure of the alveoli. We're talking about the tissue structure has a thin wall, and that's a tissue, not talking about a cell wall. Okay, doctors measure the health of lungs by calculating the FeV1 ratio to the FVC ratio. FeV1 is the maximum volume of air exhaled in 1 second. FVC is the maximum volume of air exhaled in one breath. The minimum FeV1 to FVC ratio, of healthy lungs is 0.7 to 1. A man with a lung disease emphysema inflated his lungs fully and then he exhaled as much air as quickly as possible in one breath. Figure 3 shows how the volume of exhaled air changed during his breath. So we've got time along the bottom and then volume of air in decimeters cubed up the side. So we can see over time it was increasing. Okay, use the information provided to determine the FeV1 FVC ratio of this man's lungs. Go on to determine how many times greater the minimum ratio of healthy lungs is than this ratio. So the first step here for 1 mark, and it's one of those biology hard maths questions, the first step for 1 mark is to determine his FeV1 FVC ratio. So we know the ratio of a healthy person is 0.7 to 1. Let's find out what his ratio is. So we want to check out his FeV1. And what does the 1 stand for? 1 second. So let's look up how much air did he breathe out in 1 second. And let's write that down. So we're going to draw a straight line up. And I think that goes up to there. Now, how much is each square worth? Well, there's 10 there. I think it's 2.2 decimeters because each of the baby squares, there's 10 divided by 10. Each square is worth 1. So it's 2.2 decimeters. As far as I can see, it might be like 2.15. I haven't zoomed in enough. Could be 2.15 there. And it goes up to 3.6. So his FVC, the total amount of air he can breathe out in a whole breath, is 3.6. So we now know that the ratio is 2.2 to 3.6. But notice how the original ratio was 0.7 to 1. So we need to make the 3.6 1, right? Because we need to get it into the same units. 0.7 to 1, we need to be to 1, something to 1. So we're going to divide both by 3.6. 3.6 by 3.6 gives us 1. So we now know that the FVC is 1, and we have to do 2.2 by 3.6. And so we're going to end up with a ratio of 0.61 to 1. So it's a smaller ratio. So how many times greater is the ratio of the healthy lungs than the unhealthy lungs? And so in order to find that out, I need to divide the healthy lungs, the bigger number, by the smaller number. Okay, and the healthy lungs was zero point 0.7, so I need to do 0.7 to 1 over 0.61 to 1. And if we do 1 by 1, we get 1. And if we do zero point 0.6 by 0.61, we'll get how many times bigger it is. And the answer is... So the answer is 0.7 divided by 0.61 equals like 1.1475 on my calculator, and I'm going to round that to 1.15. That's my answer. So healthy lungs have 1.15 times the FEV1 FVC ratio than unhealthy lungs. And that's worth 2 marks. Pat yourself on the back if you got that one right. These biology maths questions are a beat. Okay, let's get on with 2.3. So tidal volume is the volume of air inhaled and exhaled during a single breath when a person is resting. The tidal volume in a person with emphysema is reduced compared with the tidal volume in a healthy person. Suggest and explain how the reduced tidal volume affects the exchange of carbon dioxide between the blood and the alveoli. So tidal volume is the volume of air inhaled and exhaled during a single breath. So if we expel less carbon dioxide because they have a reduced tidal volume, so they get rid of less carbon dioxide, this means there's still a higher than really desired concentration of carbon dioxide in the alveoli. So there's less of a difference between the carbon dioxide concentration in the blood and in the alveoli. We say the concentration gradient is less steep, or it has a reduced concentration gradient, right? It's less steep, shallower. And that means we get a reduced rate of diffusion of carbon dioxide, leaving the plasma of the blood into the alveoli. And therefore, there we go. So less CO2 expelled from the lungs, 1 mark, less steep concentration gradient, a lower rate of diffusion of carbon dioxide from the blood into the alveoli for the 3rd mark. Lovely jubbly. Right, 3.1. In taxonomy, an organism is identified by referring to the species name and the genus name. What term is used to describe this method of naming organisms? So species and genus, we use 2 things, or genus and species, I should say. 2 means by, or 2, yeah. So it's called the bi-naming system, the 2 naming system, the 2 name system, binomial. So it's the binomial naming system. You can just put binomial. Define the term mutagenic agent. So a mutagen is a substance that causes changes in your DNA or your genes. So it increases. Now, you do get random mutations in your genes, but this is increasing the rate of mutation in your DNA. And there are various substances can do that, like tar in cigarettes. All right, 3.3. Figure 4 shows how the species Spartina townsendi is produced. The number of chromosomes in the cell are shown in some of the boxes. So we got the Spartina Alton Flori. I can't really read this because I'm blind. So the diploid cell is 62. Of course, what happens when you make a gamete? Think about humans when they make a sperm cell. It's got 23. It's got half the chromosomes of the diploid. So we're going to do the same here. The flower is going to have 31 in its gamete, and then it gets fertilized and it creates a new organism, Spartina townsendi, with 61. So it must have combined with a gamete that had 30, because 31 plus 30 makes 61. And that means that came from a diploid cell that was 60. And that is the Spartina, I can't even read that, maritima. Okay, a mutation in the number of chromosomes in S. townsendi cell produced a new species, Spartina angelica. Figure 6 shows the number of chromosomes in leaf cells of these species. So the S. townsendi had 61, and the new one that it creates has 122. Name the type of mutation that changed the number of chromosomes in S. townsendi to produce S. angelica. Explain your answer. Why don't you pause and have a go? Or I'm going to go through the answer. So this is known as non-disjunction. And what essentially has happened is during meiosis, the chromosomes have not properly separated. So they've not separated at all. And sometimes the homologous pairs don't separate. Sometimes the chromosomes, the chromatids don't separate. So what you can end up with, and in this case, what has ended up is that the chromosomes have doubled during the interphase, but then they to form all your pairs, but then they have not separated at all, and we've ended up with all the chromosomes in one of the daughter cells because of non-disjunction. Okay, let's move on. 3.6. Genetic variation within a species is increased during meiosis by crossing over and independent segregation. You need to know that, so flashcard that. Apart from mutation, explain one other way of genetic variation within a species. So, your main two ways that meiosis causes variation is crossing over, you get the chiasmata, to the swapping of alleles, and independent segregation, how they line up in a random order on the equator. But we can also get random mutations. And then what I'm drawing here is fertilization. Now, we don't know which sperm is going to meet which egg. So each time a sperm meets an egg, it's random. There's like 200 million sperm, one egg. And we don't know which egg has been released from the ovaries, from the millions of eggs in the ovaries. So we literally, every time we get fertilization, we get a completely new combination of alleles. And we call this the random fusion of gametes, which allows for new combinations of alleles every time. And that's going to get us 2 marks. Lovely jubbly. I think there's 200 million sperm in a teaspoon. But yeah, something weird like that. Okay, 4.1. Give two structures found in all prokaryotic cells and in all eukaryotic cells. So we're looking at structures here. So we could go with cytoplasm, although don't spell it like that unless you're a dipstick. So cytoplasm. And what else? Ribosomes. Remember, prokaryotic have 70S and eukaryotic have 80S. Now, we might say to ourselves, is cytoplasm a structure? Well, yeah, it's a fluid, but it contains lots of proteins and glycogen and enzymes and things. And of course, the cell membrane. We find that in both, don't we? Both have a bilayer with phospholipids. Right, all prokaryotic cells contain a circular DNA molecule, and some prokaryotic cells contain plasmids. Scientists have found that the rate of plasmid replication is faster in cells growing in a culture with a high concentration of amino acids than in a culture with a low concentration of amino acids. Suggests one explanation for the faster rate of plasmid replication in cells growing in a culture with high amino acid concentration. So if we have lots of amino acids, what can you make more of? Link this together. More amino acids mean you can make more proteins. Now think to yourself, how could that protein help in replication? What sort of proteins could be used? Well, for instance, in replication of DNA, we normally have DNA helicase. I'm not sure whether it's involved in prokaryotic replication, but certainly we'll get more enzymes that are involved in replication, we can say. So, yeah, for example, in humans, that'd be like DNA helicase and DNA polymerase. And I'm sure in prokaryotes, they use DNA polymerase, right? If they're making plasmids, and maybe they use DNA helicase, although the plasmids aren't in a helix, so it seems like that may be something you've got to check on the mark scheme. But I would essentially say more amino acids means more proteins, 1 mark, and then it can construct more enzymes needed for replication. Right. As scientists prepared a culture of bacterial species, she extracted plasmids and circular DNA molecules from the sampler cells taken from this culture A. She then added an antibiotic X to the culture and let the cells divide for 4 hours. She then extracted the plasmids and the circular DNA molecules from a sample of these cells called B. So B is the one with the antibiotic. A was the one they did before adding the antibiotic. She then extracted the plasmids and the circular DNA. Yeah, we did that. Scientists separated the plasmids from the circular DNA molecules in A and B using ultracentrifugation. Figure 6 shows her results A and B. We got the centrifuge tube, we got the plasmids. You can see they're thicker in B than A, and we got the circular DNA molecules are the same with antibiotic X and without antibiotic X. What can you conclude from figure 6 about the structural difference between the plasmids and the circular DNA? And then explain your answer. So the structural difference between the plasmids and the circular DNA. What is the difference here? So I would say if you're struggling with this one, just point out the obvious difference. So you can see the thick. What can you say about the plasmids? Sorry, I was going to talk about the thickness. What can you say about the plasmids in relation to the circular DNA in both tubes? So in both tubes, the circular DNA is where? It's at the bottom of the tube. The plasmids are at the top. What does that tell you about the density of the plasmids in comparison to the density of the DNA? It doesn't really tell you about the amounts. This is about the density. So it tells us that the circular DNA is more dense because it appears lower down the tube. What can you conclude from figure 6 about the effect of antibiotic X on plasmid replication and on circular DNA replication. So if we look at antibiotic X, this is the one that was taken later, right? So first of all, they did the sample A, and then they added the antibiotic and did sample B. So it's taken later on. We can see that the circular DNA did not increase. It didn't get fatter, the band, from A to B. And that means that the replication of the circular DNA has stopped. Antibiotic X inhibits replication of the circular DNA. However, did it affect the plasmids? No, because when we look from A, which was before, without the antibiotic, then we look after, we can see the plasmids have got bigger, which means plasmid replication is continuing. So antibiotic X inhibited the replication of the circular DNA, and we can see because the bands have remained the same thickness. However, the plasmid bands has gone thicker, and this is because antibiotic X allowed the replication of the plasmid to continue.
Okay, question 5. A student investigated the activity of the enzyme amylase. He cut 3 identical wells, D, E, and F, in starch agar in a petri dish. He added 0.2 centimeter cubed of amylase solution to well D, boiled the amylase solution to well E, and only added water to well F. So we've got one well with amylase, that enzyme. We've got one with boiled amylase, that's well E, and then we've got one that's got no enzyme, it's just got water in well F. After 60 minutes, he covered the starch agar with iodine. And figure 7 shows the results. So we can see it's all gone blue-black. What does that tell us the presence of when iodine goes blue-black? Except for around well D, where we've got a clear area. Okay, moving on. Explain the appearance of the agar in the clear area around well D. Well, should be fairly easy, right? So grab a pen and paper, have a go at the question. Okay, I'm going to go through the answer. So obviously, we can tell that amylase hydrolyzes, you could say breaks down, but they might not accept that, or catalyzes, which I can't spell, so let's leave it at that. Catalyzes or hydrolyzes the starch. So no starch is present. So there's no starch left over. I hate spelling. I'm typing. It's typing I'm rubbish at because I've got fat fingers. So no starch is present. So it does not turn blue, black. There we go. Oh, that was long. Basically, there's no starch there because the amylase has hydrolyzed or catalyzed all of it. Okay. Let's go down to 5.2. What can you conclude about the activity of amylase from the appearance of the agars surrounding well E and well F? Well, we can say the amylase has been denatured in well E. So the amylase has been denatured in well E as it was boiled. Yeah, it can no longer form a, substrate bloody enzyme substrate complex as it was boiled. Okay, so amylase has been denatured, hence no starch is hydrolyzed. In well F, it was only water. There wasn't any amylase. So we can say well F shows that the starch does not hydrolyze in just water alone. It shows amylase is needed. So not only does it proves that amylase is needed and that the starch doesn't just break down on its own if you leave it. So it proves that with well F, and well E proves that the amylase mustn't have been boiled. So if it's boiled, it gets denatured. So, okay, let's just give ourselves, oh my gosh, look how I spelled denatured, then Harold. Anywho, right, let's move on. 5.3. The student cut a piece of agar from the clear area around well D. Okay, he obtained a solution of the substances contained in the piece of agar. Describe a different biochemical test the student could use with this solution to confirm that amylase had affected the starch in the clear area around well D. So if we look up at well D, what they're saying here for this question is that the student cut a piece of this clear area out. Now, there's no starch there. But what would confirm that the amylase has hydrolyzed the starch? What would you expect to be there in this area? So obviously, when starch breaks down, what do we make? We make glucose or maltose. We make simple sugars like maltose or glucose. And so what test could you do to confirm this? So we would use heat the sample with Benedict's reagent or Benedict's solution. Um, it will go from blue to, what do we reckon, a red precipitate. You probably can say orange, yellow. You could probably say green, orange, yellow, or red, or green, yellow, orange, or red. Any of those you can say, because they're all the colors that it'll go. Cool. And that would confirm that glucose is present, or simple sugars like maltose and glucose, reducing sugars. Okie dokie. The diameter of the clear area around well D is 18 millimeter. So what they're saying is that the area around D, this is D, they're saying the diameter is 18 millis. Okay. In a different investigation, the student prepared a solution of the amylase solution. He did this, or a dilution of the amylase. He did this by mixing the amylase solution from well D and water in the volume shown. So 1.6 amylase, 2.4 water. He prepared a starch petri dish identical to figure 7, but with a single well. He added 0.2 of the diluted amylase to the well and left the petri dish for 60 minutes. Use this information to predict the diameter of the clear area to form around the well containing the amylase solution. So we know that when it's 100% amylase, it's 18 millimeters. What is the percentage of amylase now? And then do that times that. So the total volume here is 4 centimeter cube. And we want to find out what percentage of that is amylase. So it's 1.6 over 4. I'm not exactly sure. Where's my old calculator? Is that 0.4? What is that? It's definitely less than 0.5. It's 0.4, yeah. So it's 0.4 or 40% amylase solution, and 100% amylase made that. So what would 40% make? Well, we go 0.4 times 18, and it wants it to the nearest whole number. I got like 7 point something, whatever that is. Let's bash it in the calculator just to do it. So times 18, 7.2, but we're going to go to 7 millimeters. There we go. So lovely jubbly. That would be the diameter we would expect, 7 millimeters. Super duper. 2 marks. That one wasn't too bad for a biology maths question. Okay, 5.5. The student used a ruler to measure the diameter in millimeters of the clear area around well D. Use this information to explain why the answer to 5.4 should be given to the nearest whole number. Well, because if they're using a ruler, that will measure in millimeters. Millimeters will be the smallest division on the ruler. So it's likely, unless they say that there's, I mean, I've not seen a ruler that goes to like 0.5 millimeter or 0.5 millimeters. So normally a millimeter is the smallest division on the ruler. So you would measure to the smallest scale that your ruler can measure to. So that would be how I would answer this question. Okay. Question 6. The fruit fly is a species of small insect. The fruit fly has a gene that codes for the enzyme called alcohol dehydrogenase, AD. AD catalyzes the breakdown of alcohol when alcohol is in the insect's food. The gene coding for AD has 2 alleles, ADF and ADS. The enzyme encoded by the ADF allele causes the breakdown of the alcohol faster. I guess the S is slower than the enzyme encoded by the ADS allele. Suggest why. So why is it that the this allele can basically catalyzes the breakdown of the alcohol faster? Well, it's not the allele that does it itself. What is it that will be breaking down the alcohol? It will be the alcohol dehydrogenase enzyme. Now, what is it, about the alcohol dehydrogenase enzyme that's coded for by ADF that could be different than the alcohol enzyme coded for by ADS? Why is it that ADF works better? So what I would say is it's got a different, possibly a different primary structure, and therefore if it has a different primary structure it will have a different tertiary structure. So we might want to say different amino acid sequence or primary structure. So it codes for a different I might want to be a little bit more fancy. So ADF codes for, it won't be very different, but a slightly different amino acid structure or sequence or primary structure. This leads, due to the different ionic bonding, covalent bonding, etc., leads to this leads to a different tertiary structure, and so a different 3D shape. The active site makes a better, or let's use better words, A-level words, more complementary fit with the substrate, which is the alcohol, obviously. And so it breaks it down, enables it to weaken the bonds and break it down faster. Anyhow, I think that'll be enough for 3 marks. Basically, different primary structure gives you a different tertiary structure, meaning the active site has a better fit or a more complementary fit with the substrate. 3 marks. That's what I was essentially getting at. Okay. Ah! A scientist took a random sample of adult fruit flies from a population. He measured the frequency of the ADF allele in the sample generation 0. He then sealed 100 of these insects at random and kept them in a container, fed the insects food containing alcohol, let the insects reproduce, repeated this 45 generations of fruit fly reproduction. The scientist measured the frequency of the ADF allele in the 45th generation. Suggest why the scientist took the samples from the population at random. Well, the word random, you know this one, it's to avoid bias. You know that one. That's why we do random samples. It avoids bias. Okay, table 2 shows the scientists' results. So in generation 0, the frequency of the ADF allele was 0.2. By the time they got to generation 45, so 45 generations of little fruit fly children later, the frequency of the ADF allele is 0.74. Alcohol is toxic to fruit flies. We've got a selection pressure here. Suggest and explain why the frequency of the ADF allele changed during the 45 generations. So what is going to increase the frequency of the allele down to this selection pressure that flies that take the alcohol and can't break it down will die. So 4 marks, it's basically natural selection. Have a go, pause the video. All right, I'm going to go through the answer to this. So essentially, this is your natural selection bit. So we want to say mutation, or let's just say, let's skip the mutation, but the mutation that gives flies the ADF allele means they can break down, I should use the word hydrolyze, but I'm not going to go back, break down the alcohol faster. So they are more likely to survive. You know, this as survival of the fittest, more likely to survive. If they're more likely to survive, what are they more likely to be able to do? So they are able to breed, so more likely to breed or reproduce, pass on the ADF allele to their offspring. And then we want to say over many generations, well, 45 exact, but over many generations, the frequency of the ADF allele increases. Now, when you do natural selection like this, you get a natural selection question. It's almost the same as at GCSE. At GCSE, you would say whatever the beneficial advantages from the mutation makes them more likely to survive, makes them more likely to breed, pass on the gene or the allele to their offspring. But the A-level bit that's always key at A-level is that you have to say the frequency of the allele increases, or increasing the frequency of the allele in the population. I'm probably just going to end that with in the population, just to be a bit more specific. And here we go. 6.4, identify the type of selection investigated in the 45 generations of fruit fly reproduction. So what we're looking at is it started off at 0.2 and it ended up at 0.74, right? If we look at the table of results. So it's definitely the selection has gone in where it's gone from one to one of the extremes. So if it had gone down from 0.2 to 0.01, then it's gone to the other extreme. But as it's gone, increased the frequency quite dramatically from 0.2 to 0.74, we would call this directional selection. So it's gone in a direction. Stabilizing selection would be that it stays in roughly the same place. And random selection, well, random selection, we would have had the frequency of the allele change, but not by much, because it would just be by the random nature of breeding and stuff like that. So, yeah. Anywho, Okay, so describe an ATP molecule. No, describe how an ATP molecule is formed from its component molecules. So we really want to say, what is the structure of an ATP molecule? And that will get us some of those marks. And then how is it formed? I.e., what is the reaction that forms it? So let's go ahead and draw an ATP molecule so that I know what the hell I'm talking about. Right, what's that? This is adenine, and you have that is that's a weird looking adenine, people. Okay, so we've got adenine, and then does it come off of there? I think so. Probably done that completely wrong. So you've got adenine, and then we've got a ribose sugar, and then we have three phosphates. And so that, I'm not really happy about that drawing. It looks a bit rude, but yeah, what are you going to do in life, eh? Okay, let's write this out. So it contains adenine, a ribose sugar, and three phosphate groups or molecules. Okay, so that's what it contains. Now, it is formed in a condensation reaction. So it is formed in a condensation reaction. What else can we say? Isn't it between ADP and PI? From ADP and PI. And what enzyme puts the ADP and the PI together? I'm going to say by ATP, whoopsie, not by ATP, whoopsie, but by ATP synthase. And that's going to get us 4 marks. So 2 marks for describing the molecule, then saying it's formed in condensation ADP and PI. I'm not sure if you need the condensation or the ADP and PI, but I'm going to shove those both there. That one of them is going to get me that 3rd mark. And then by the enzyme ATP synthase, that is how it's formed from its component molecules. I could say removing and releasing a water molecule, just shove that in there as well. You never know when they're going to put something like that on the mark scheme. There we go. Orsomenous. Right, let's zing down. A scientist investigated the effect of cyanide on the rate of amino acid uptake in 2 types of Escherichia coli. So that means E. coli. Okay. G cells produce enzymes involved in ATP production only on the cell surface membrane. H cells produce enzymes involved in ATP production only on the cell surface, no, on the cell surface membrane and the cytoplasm. So H does both. Cell surface membrane, it makes ATP and in the cytoplasm, whereas G cells can only make ATP on their cell surface. We've got the rate of amino acid uptake against the concentration of cyanide in G and H cells. We can see here that the H cells, which can do it on their membrane and their cytoplasm, as you increase the cyanide, yes, they're not able to take in amino acids as well, but they get to a point where they can always take some amino acids in, even if it plateaus there. Whereas here, the rate of amino acid uptake in these cells, which only make ATP on the cell surface membrane, it drops rapidly and then it goes non-existent at about 18 millimole per decimeter of cyanide. Okay, let's get on and have a look at the question. Use figure 8 to calculate the percentage decrease in the rate of amino acid absorption by H cells, not H and G, just H cells, in the 30 millimole per decimeter cyanide solution. Let's go and check this bad boy out, right? We're going to do this up here. So we're looking at the H cells, the solid black line. I want to zoom in a little and make that a bit bigger. No diddy. Right, there we go. So we've got this solid black line here. And what we want to do is just check, 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 check. What did they say? Oh my gosh, I've already forgotten. Was it 10 or was it 20? All my days. It's not going to be anything as simple as that. Let's go down. 30 millimole cyanide H. Okay. So at 30, we are looking at what's that? 1.2 rate of amino acid uptake at 30. And then from the start, what was the start here? Well, each one of these squares is worth 1. That's quite easy to work out. 10 squares and the value is 1. So each one is worth 0.1, sorry. So that is going to be 2.7, 2.8. So I'm really looking at the difference, which is 2.8 minus 1.2. We want the percentage decrease. So we're looking at 1.6 over the original. What's the original? 2.8 times 100. Bang, bang. Should be dead easy, that, right? What is the percentage decrease? 1.6 divided by 2.8 Is it 2.8? Yeah, it is 2.8 equals 0.5714 times 100. Let's go 57.14. So 57.14%. Boom. We could leave that at 57.1. I'm not sure if the mark scheme just wants you to put 57, but they should accept 57.14, and that'll get you two fat marks. Another biology maths done. Let's go. Using figure 8 and the information provided, what can you conclude about the amino acid uptake by G cells and H cells? Well, look, what we can tell, we can conclude that cyanide doesn't tell us about what cyanide does. So the rate, it doesn't really tell you what cyanide does, but cyanide inhibits respiration. So cyanide actually inhibits respiration. It's a inhibitor of some of the enzymes involved in the production of ATP. So the fact that amino acid uptake goes down when you increase cyanide concentration tells us that amino acids are taken in by active transport, because when you can't produce ATP, you can't take the amino acids in. So one of the things that we can tell is amino acid uptake uses active transport. So that's one thing we can tell. Amino acid uptake uses active transport. Less ATP means less amino acids taken in, or yeah, you get the idea. Anywho, we've already kind of banged on about that point. That's going to get us a mark. What else do we need to say here? Aha. ATP production. What one did it disappear in? It disappeared in the G cells. So ATP production in G cells is only on membrane. That's cool. ATP production in the G cells is only on the membranes. As amino acid uptake falls to 0, it shows us the enzymes for ATP production are only found on the membranes, right? Because if they had enzymes that can make ATP in the cytoplasm, then yeah, it wouldn't have. Or it shows us that cyanide inhibits the enzymes on the membrane. So cyanide inhibits ATP enzymes on membranes. However, as amino acid uptake continues in H cells, this shows that cyanide does not inhibit the ATP enzymes in the cytoplasm. At least, maybe not. Yeah. Well, it's showing that ATP production in the cytoplasm is continuing, because the other one only has ATP production on the membrane, so that's inhibited, but the cyanide doesn't inhibit the ATP production in the cytoplasm, which is why the H cells can keep going. Okay, we're doing question 8. A scientist investigated a sequence of reaction catalyzed by two enzymes, GOX and HRP. Figure 9 shows this sequence of reactions. Here's figure 9. We can see we've got a glucose molecule. GOX is an enzyme. It's helping hydrolyze or catalyze or whatever, turn this glucose molecule into gluconic acid. We've got oxygen going to hydrogen peroxide, and then the HRP using the hydrogen peroxide as a substrate to make water. Then we've got a colorless pigment also being catalyzed by the HRP into a green pigment. Let's look at question 1, 8.1. Use figure 9 to identify all of the products formed when this sequence of reactions is completed. Why don't you try and just name these in your head? Pause the video, then unpause, and I'll go through it. Okay, well, we can see what the products are. The products are at the end of all of these. So this is the substrate, this is the product. Oxygen's the substrate here with GOX. Hydrogen peroxide's the product, but then it acts as a substrate for HRP, which is finally converted into water. So you really want to look to the end of the arrows, where the arrows end, to find the final product. And this colorless pigment is the substrate and being turned into this green pigment. So we want to write those three down. So I'm not going to bother putting them down because I've circled them. So gluconic acid, water, and the green pigment. Right, let's move on. We don't want to waste too much of your precious time. Okay, we've got a biology 3 marker and it's a maths question. 8.2, the scientists joined DNA molecules together to make tiny cages, kind of like nano cages, I guess. The cages are exactly 20 nanometers long, 20 nanometers wide, and 17 nanometers deep. What does this make you think you're going to have to calculate if you're given the length, width, and the depth? Anyway, he trapped one GOX molecule and one HRP molecule together in each cage. So we've kind of got a cage. I like to draw things out, right? We've got a cage and we've got a GOX and we've got an HRP inside the cage. Right, where were we? The GOX molecule and the HRP only fill 9% of the cage's volume. The volume of a GOX molecule is 8 times, this is 8 times larger than the HRP. So they're kind of in a ratio of 8 to 1. Use this information to calculate the volume of one GOX molecule and give the appropriate unit. Show you're working because you want to get some of those marks if you can't get them all. Why don't you grab a pen, grab a calculator and a paper, have a go. All right, listen, unpause, whatever. I'm going to go through the answers. Okay, so what we want to look at, first of all, it should strike everybody. Can pick up 1 mark here by just working out the total volume of the cage. So our cage has a volume of 20 times 20. What's that? 4,000 times, no, 400, whatever, times 17. My mental maths is shocking. 20 times 20 times 17. Yeah, 6,800. So I believe that is going to be your first mark. Now, what would the unit be? Well, you've done nanometer by nanometer by nanometer, so it's going to be nanometer to the 3 because you timesed it 3 times. Okay. Right. What next? What next? What next? Right. 9% of this is the GOX and the HRP. So let's times that by 9%. So times 9 over 100. If you can do that in your head, you'll realize that it's 0.09. So let's times that by 0.09. Smash that in the calc. Times 0.09. That equals 612. So we now know that the GOX and the HRP are 600 and, oh, what is that? 612 nanometers is the volume of the GOX and the HRP. They're in a ratio of 8 to 1. So we could really do 612 divided by 9, because the total of that is 9. We're splitting it into 9 parts and giving 8 to GOX, 1 to HRP. So we could do 612 divided by 9. That's going to be, oh my days, what is that? Divided by 9 is 68. So that means that there are 9 lots of 68. So we can do 8 times 68. And that's going to get you your GOX volume. So times 8 is 544, like that. That is the volume of the GOX. Now, that's going to also be in nanometers cubed, and that's going to get you 3 fat marks. You can always test it out and be like, oh, I'm not sure I've done the right thing. I'm not sure I've done the right thing. Well, just divide that by 8 and you'll get 68, which is 1/9 of that. So, and it makes sense, right? If the HRP, if GOX is 544 and HRP woohoo is 68, and then you go, right, let me do 8 times 68 and you get 544, you know that that is 8 times bigger than that, 8 to 1. There we go. Right, what will you get the marks for? Well, you get 1 mark for working that out, and then they're going to be, you're going to get 2 if they see anywhere like 612 on there. If they see 68 on there, you're going to get a mark. If they see 544, you're going to get a mark. But obviously, for the final answer, you have to, if you want the 3 marks, you need to say nanometers cubed because it specifically asks for the unit. So yeah, there you go. But you can pick up 2 marks just from working out the overall volume than any of these other key numbers on the way. All right, let's move on. The scientists investigated the activity of GOX and HRP enzymes when they were trapped in cages, T, and not trapped in cages, NT, but free in solution with no cages. So no cages is NT and in cages is T, trapped. All right, so trapped and not trapped. Okay, figure 10 shows his results. The error bars show plus on minus two standard deviations and include 95% of the data. When we see 95%, we go, okay, this is quite reliable. Um, Statistically, we can talk about significance here. Let's see what the actual question is. What can you conclude from figure 10 about the effect of trapping GOX and HRP inside the cages? And it's worth 3 marks. Let's do it up here, where the actual graph is. So the relative activity of GOX and HRP is higher in cages. So we want to say relative activity of hormidase HRP and GOX is higher in cages. We can see that clearly there. The fact that these two bars, look, there's no overlap between the bars. They don't overlap. There's a space, is we can say it's significant. It is a significant difference. And then for the third mark, we give the reason why as no overlap between error bars. Okay, so there's no overlap between the error bars. Let me rub out those two dodgy black lines. Okay, you could word this slightly different. If you prefer, you could say the relative activity of HRP and GOX is significantly higher in the cages than not in the cages. And this is shown by the fact there's no overlap between the bars. So, yeah, that's what we could say. And that get you 3 lovely marks. Right, let's move on. Now you might say, oh, you remember this was the word conclude. If it was evaluate, then they'd be telling you there's something wrong with the experiment and there might be evidence going for both ways. But we didn't need to go looking for that because they didn't say evaluate. So exam technique is important and understanding the command words. 8.4, the design of the scientist's investigation did not include a suitable control. So there's something which isn't so good. Suggest a suitable control. When you're asked to suggest a control, you should always ask, what is my control trying to prove? And we've got a situation here. We've got a situation where we have the enzymes basically loose in fluid, and then we have the enzymes trapped in a cage, right? They're trapped in a cage. We found that the activity was significantly higher. What should a control be? Well, we want to show why, when the enzyme was in the cage, was it higher? Was it because of the enzyme being in a cage, or is it just because of the cage? Maybe it's nothing to do with the enzyme. Maybe it's the cage, the DNA cage, that's causing higher activity. So what we should do as a control is just get an empty cage, or a cage with denatured enzymes in it to prove that it's not the cage that's having an effect on the relative activity. Okay, so there we go. Yeah. That would be empty cage or a cage with water in it. So you can check whether it's the solution, or a cage with denatured enzymes in it. Okay, let's get on to the last question. 9.1, explain 5 properties that make water important for organisms. Right, explain. So you're not going to get any marks for just describing the 5 properties of water. You're going to need to explain a reason behind them, but it's only 5 marks, so you can't write a whole essay on it. You just want one succinct bullet point reason to explain each of the properties. So, for example, water has a high specific heat capacity. Now, I could explain what that means. This means that a lot of energy is required to raise the temperature of water even by 1 degree, but we want to explain why it's important for organisms. And we can say it resists changes in temperature, or it buffers changes in temperature, or we can say resists, buffers or resists is a good word. All my days. So That's why it's good for organisms. Okay, we don't have to go into all this is good for marine life living in the water. We don't have to go into detail. It's not an essay. Right, another one is now we're on high specific heat capacity. It has a high latent heat of vaporization. So high latent heat of vaporization. How does that help? Well, a lot of energy is needed to make the water, the sweat on your body evaporate. It requires a lot of energy to do that. So when those molecules leave your body, they carry away with them a lot of heat energy, and this has a cooling effect on you. Now, we don't need to put all of that fatness down. We can just say high latent heat of vaporization means it has a cooling effect, or we can say it's a very good coolant material. Okay. And the reason why it's a good coolant material, well, these are only be 1 mark each because this is super stingy. Whoopsie. Right, let's do another one. We can talk about cohesion. Again, this is down to the fact that water molecules have a dipole, but we're just going to say water molecules have cohesion. We can say cohesion tension if we want. And why is that useful that they have cohesion? This means the water molecules can stick together via hydrogen bonding and can be drawn up the xylem by cohesion tension, should be a hyphen in between those, in a continuous water column. Now, do I need to write all of that? No, that is just going overboard. We just need to say water molecules have cohesion. This means they can form a water column, which is useful in plants, right? I've just gone a bit overboard. Okay, so that looks like it's 4 marks, but it's not. That's only I want to get rid of that little tick. Oh, jeez, Louise. All right, you're just gonna have to, yeah. Okay, so that tick is for all of that explanation. So we're up to 3 marks now. You can see this was quite a difficult question because there's a lot of writing just for 5 marks. Okay, so we've done cohesion, we've done, oh, we've got water as a solvent. So water is a good solvent. Well, it's a good solvent for polar metabolites, such as glucose and amino acids. Yeah, so it's a great solvent for things like glucose and amino acids. So I could have just said it's a good solvent. Why is that useful? It's good for dissolving glucose and amino acids, which are important in the body. Oh, water's also good. Water is needed, talking about little molecules, it's needed in metabolic reactions. Um, so water is a metabolite in condensation. Can't spell it, guys. Can't spell it. Condensation and hydrolysis reactions. We could have got specific if you're an A-level student here, you could bust out photolysis or something like that. Um, Yeah. So there's 5. What other things? I could have gone on about surface tension. So that the water molecules are polar, they can hydrogen bond, and therefore do surface tension on the top of ponds and lakes. And that allows insects and things to walk across the water. Let's move on. Bada bada bing. Right, 9.2, describe the process of semi-conservative replication. Key word here is we don't have to explain, guys. You've just got to say this. It should be dead easy. 5 bullet points. Go for it. Pause the video, have a go. All right, I'm going to go through. It's easy peasy. So we're just going to say DNA. Do you remember the enzyme? Helicase breaks the H bonds. Oh my gosh, check out that O between the strands. Or we can say unwinds. So that's optional. You could say unwinds the DNA strands or unwinds the polymers. So those really would get you the same mark. You don't have to write all of that. What can we say next? Let's go for the other enzyme, DNA polymerase. helps join free DNA nucleotides. Now, AQA are quite generous. You can just say free nucleotides. You don't have to say free DNA nucleotides, but some examples like OCR are specific on that. So DNA polymerase helps join free DNA nucleotides to the template strands. So the template strands, yes. And how do they do this? We can say by complementary base pairing. Now, if you said complementary base pairing, you don't need to put this next bit. But let's say you're in an exam, you're like, oh, I can't remember that term. Then you can just say like A with T and G with C. Now, I highly recommend that you don't do what I've done there. I highly recommend that if you can't think of complementary base pairing, you say adenine with thymine and guanine with cytosine, and you actually spell them out because sometimes on the exams they will not allow just random abbreviations unless they've been mentioned in the question. So you can't always do that. So we're going to get 1 mark for DNA helicase breaks hydrogen bonds or unwinds the DNA. 1 mark for DNA polymerase helps join free DNA nucleotides to template strands. Um, so lovely jubbly. And then what we want to say is, so that would be 4 marks already. So 1 for DNA helicase, breaking the hydrogen bonds or unwinding the DNA. 1 for DNA polymerase, joins free DNA nucleotides to the template strands. So those are all key points. Via complementary base pairing is a mark. And then what we need to say is phosphodiester bonds, people. So there's a condensation reaction. Uh, condensation reaction forms, and again, it's the DNA polymerase, forms phosphodiester bonds between the nucleotides. So the backbone gets fully formed then. And then finally, you could say, as it says here, specifically semi-conservative, you could explain that the daughter strands or the new strands um, are made up of one template, or let's go with one original strand and one new strand. I shouldn't say the new strands then, should I? I should say the new DNA. I prefer to say the daughter DNA is made up of a parent strand and the new strand or a daughter strand, but yeah, whatever. All the same thing. That'd get you a mark. So there's 6 possible marking points there, and we only need 5. All right, guys. Don't forget, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you lot on the next one.
You're still here? It's over.